Hi, everyone. Welcome to our second round of genetics labs. So to date, what you should have accomplished for me is you should have done an online lab module using the Labster platform, and that covered polymerase chain reaction as well as gel electrophoresis. And then for your in-class portion of your lab assignment, you should have completed the activity where we used gel electrophoresis along with restriction fragment length analysis to make a diagnosis about sickle cell anemia in a family. So you should have accomplished those things. You should have those things turned in to me. This week, we are going to deviate from our plan a little bit. I actually did plan to do this activity with you as part of our online activities, but we are going to all be online this week because I have to take a relative to a medical procedure. So today, what we're going to do is we are going to use something called branch diagrams to solve some genetics problems. Now, very quickly, probably toward the end of this week, in Dr. Bauer's lecture, you are going to begin a unit on what we often call transmission genetics. In other words, how on earth do we go about transmitting traits from one generation to the next? You may also have learned this as what's often called Mendelian genetics, because the field of transmission, Mendelian, I can't talk and write, Mendelian genetics. The field of transmission genetics was pioneered by a monk named Gregor Mendel, who was working in the 1850s and 1860s, and he really helped to define some of the rules about how traits are transmitted from parents to offspring. Now, you probably have some familiarity with this already from high school or from your general biology classes. You have used the basic rules of Mendelian genetics and you have plugged those into something called Punnett squares, right? And we're going to review this just in case this isn't ringing a bell for you. But in the past, you, I know, have used some knowledge of genetics and you have used something called a Punnett square to understand how a parent might mate with a second parent and produce a suite of possible offspring. Today, I'm going to show you another method for solving genetics problems called the branch diagram method. And this method is particularly useful when you're dealing with more than one gene at a time. So let's back up for just a second and review the basics of Mendelian genetics, okay? So I mentioned that Gregor Mendel was this monk. He was working in the 1850s and he made some foundational discoveries. First of all, he discovered that traits were passed on by factors and that these factors had different forms. Okay, we later used the term gene. So traits are passed on by genes. And these genes can take on different forms, which we call alleles. What do I mean by that? Well, let's take a look at these pups over here. So this is a breed of dog called a Weimaraner, and it happens to be the type of pooch that I own. Some of you know that I have a puppy named Indiana. Yes, his name is Indiana Jones. You may call him Dr. Jones. And Indy looks like this kind of Weimaraner right here. But notice that Weims, which is what they're often called, Weims come in two flavors. They can be what is called a blue coat color, or they can have a gray coat color. Oops, come on, there we go. And so here's an example of what they look like as puppies. Here's a gray puppy here, and here's a blue puppy here. This is exactly what my Indy looks like. Indy is this one right here. He's a little blue Weim, okay? So what we can do, let's say that we have a family of blue Weimaraners that are always blue, like mom was blue, dad was blue, babies were blue. And if you go back many generations, we have a whole bunch of blue Weimaraners. So we're going to call these true breeding blue. So these are true blue Weims, okay? And let's say that we take a true breeding blue Weim and we cross it with a true breeding gray Weim, meaning that if you go back for many, many generations, it's always a gray Weimaraner and they always produce gray offspring. So we'll cross a true breeding blue with a true breeding gray. Okay, if you performed this cross, what you would find is that all of the offspring would be blue. So what does that tell us? Well, it tells us that whatever this parent is contributing to the offspring is masking what this parent is contributing to the offspring, right? This parent's contribution is covering up this parent's contribution. And you probably know where I'm going with this. One of the things that Gregor Mendel discovered is that some traits tend to be dominant and they tend to cover up other traits, which we call recessive, right? You guys remember this. So in Weimaraners, blue coat color is dominant to gray coat color. Now, let's take a look at this cross and let's use a Punnett square to describe what happened here. So if you remember, a Punnett square looks very simply like this, okay? Got a square, and we're gonna make some little windows in it. Okie doke, y'all have seen this 100 times. Now, I told you that we had a true blue Weimaraner. Now, we know that dogs are diploid. They have 2 copies of each gene, right? And if every single Weim in this family's history was blue, then that probably means that each copy of the gene, each allele that these Weims had must have coded for blue coat color. So we're going to have big B equal blue coat color, right? Now, that was our blue. Let's talk about our gray. If all of the ancestors, if we had many, many, many generations of gray Weims, the likelihood is that what you had were a bunch of individuals that had 2 copies because they're diploid, and they probably had 2 copies of alleles that coded for gray, which we're going to have be little b because we learned that gray is recessive, okay? So we have big B, little b, little b. Now, let's just say that this is mom and this is dad. Mom and dad are only going to give one of these copies to their offspring. So what we're going to do is we're going to simulate mom can give this copy of a gene to an offspring, or it can give this copy of a gene to an offspring. So we've simulated gamete formation, okay? In other words, which gene she can put in her egg cell, okay? So she can either make an egg with a big B or she can make an egg with a big B, right? Because she has those copies. Dad, when he goes to make sperm, he can either make a sperm that has this copy, so I'm gonna put that here, or he can make a sperm that has that copy, so I'll put that here. Now, if this egg fuses with this sperm, what we have here, we've restored our diploid state, but we have one big B, one little b. This guy here covers up this guy here. And we can go through and we can do this for each possibility. So what a Punnett square shows us is the possible outcomes of a cross, right? That is what a Punnett square shows us. Now, what if we took two of these individuals? I mean, we shouldn't, they would be siblings, but let's just say that we did. What if we took a wine that had this genetic makeup and we crossed it with another wine that had this genetic makeup? All right, let's draw a Punnett square. Boom, 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 boom. All right, so let's say this is mom. Mom can make an egg that either has a big B in it, or she can make an egg that has a little b in it. Dad can make a sperm that either has a big B or he can make a sperm that has a little b. And if we perform this set of crosses, we have a different universe of possibilities. All right. Now, let's do some quick review. If you have two copies of the same allele, we say that you are homozygous. Homo meaning same, okay? You are homozygous for that allele. If you have one of each, we say that you are heterozygous. Okay, so this one's homo as well. Hopefully, this is all review. Now, I want to, let's see here, I'm going to go up here and I'm going to insert a new slide. Now, I want to go back. Hold on one second. Let me get this to come off. Nope. Stop. There we go. Now it should click off. Okay, so I want to revisit our cross we did here with two heterozygotes. Let's revisit that. So we have big B, little b, big B, little b, boop, 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 boop. Here we go. Okay, so there are four possible outcomes if these two individuals mate. You can be big B, big B. You could be big B, or you could be little b, right? Chances are there's 1 in 4 chances that you would be this. There's 2 in 4 or 1/2 chances that you would be this. And there's 1 in 4 chances that you would be this. This is often called the genotypic ratio. In other words, to get this genotype, you're 1 out of 4. To get this genotype, you're 2 out of 4. And here you're 1 out of 4. This is also the probability of getting these offspring. Probability, okay? So you have a 1 in 4 chance of being homozygous for the dominant gene. You have a 2 in 4 or a 1/2 chance of being heterozygous. And you have a 1 in 4 chance of being homozygous recessive. Now, there's another way that we can look at this information as well. First of all, let's kind of look at our genotypes that we have here, our options, and let's look at what's called the allelic relationships. Okay, so if you are big B, what does that look like? Well, hopefully you remember that individuals with this big B allele, they had a blue coat color, right? If you were little b, on the other hand, you have a gray coat color. And if you are heterozygous, because this allele masks this allele, you are also a blue coat color. So another way of analyzing this Punnett square is to simply look at the physical traits that result from this possible cross. And if we do that, we can see that this critter would be blue, this one would be blue, this one would be blue, and this one would be gray. So another way of thinking of this is that you can either be blue or you can be gray, and you have a 3 out of 4 chance of being blue and a 1 out of 4 chance of being gray. Or you could even just say a 3-to-1 blue to gray ratio. This is called the phenotypic ratio. Okay, so the genotype is the actual genetic makeup. And here our genotypic ratio would be a 1 to 2 to 1, where you have 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. Your probabilities would be 1 out of 4, 2 out of 4, and 1 out of 4. Here we have our phenotypic ratios, or our phenotypes. Your phenotypes are just the physical appearances that result from these various combinations of alleles. Our genotypic ratio would be 3-to-1, and our probabilities would be 3 out of 4 that you would have a blue dog and a 1 out of 4 that you would have a gray dog. Now, hopefully, all of this is review, and I tell you all of this because we're going somewhere with this. Let's make things a little more complicated now. You probably did a lot of work in high school and maybe even in your biology 105 classes looking at one gene, but let's look at 2 genes simultaneously. So let's look at coat color in Weimaraners, okay? And so coat color, we had the big B allele, which was blue, and we had the little b allele, which was gray. Blue was dominant to gray, and we often designate that by using an uppercase letter, okay? Let's include a second gene. Now, this is true. This is like a real thing, but the second gene I'm going to make up. Now, Weimaraners are famous for snoring. So let's introduce this fake gene here of big S, which equals a snoring dog. And I'll make my little s like that so you can really tell. That's a quiet dog. And you can tell that in my hypothetical scenario here, a snoring dog is dominant to a quiet dog because this is capitalized. Okay, so now we're going to look at 2 genes, and I want us to do something called a dihybrid cross. Now, when you see this term hybrid, I want you to think heterozygote. Okay, and if we're talking about a dihybrid cross, we must be talking about a cross between people or animals that are heterozygotes for 2 genes, di meaning 2, hybrid meaning heterozygote. So if I'm talking about a dihybrid cross, here's what I mean. We're going to have one dog that is heterozygous for coat color and heterozygous for snoring. And we're going to mate them with another dog that's heterozygous for coat color and heterozygous for snoring. Hopefully that makes sense to you. You can see that this would be a blue snoring beast because even though this is heterozygous, this blue is covering up the gray, right? And even though this is heterozygous, this snoring is covering up the quiet. Now, how on earth would you set this up? Well, the first thing that you have to recognize is that, again, we're talking about diploid organisms. So if we're talking about mom here, she can make an egg that either has a big B or it has a little b, but in that same egg, it's either going to have a big S or it's going to have a little s. So we can kind of figure out what her possible gametes might look like. She can have a big B, big S egg, right? She could have a big B, little s egg. She could have a little b big S, or she could have a little b little s. Let's do that again because that's a little bit confusing. Big B big S, big B little s, little b big S, and a little b little s. Okay, some of you may be familiar with this methodology here. In math, we call this the FOIL method, where you multiply the first terms of an expression, the outer terms of an expression, the inner terms of an expression, and the last terms of an expression, okay? So these are all of the possible eggs that mama could make, where she gives a copy of each gene to her eggs, or all of the possible sperm that dad could make, where he gives a copy of each gene to each sperm. Now, if we go to make a Punnett square, it's going to be a monster, right? We're going to have to make it look something like this. We know that mom and dad both have 4 options for what their eggs or sperm could look like. So we got to make 4 slots for each of them. 1, 2, 3, 4. 1, 2, 3, 4. And so you would put all of mom's options up here. So hopefully you can see that I just took here these options here and I wrote them across the top. Here's all of dad's options. Let's put them here. Now, within each of these squares, you would figure out what each of these possible offspring would look like. And so here's a big B, big S, or sorry, big B, big S, big S. This is a blue snoring dog. Big B, big B, big B, little s. This is a blue snoring dog. Oops, hold on, I screwed that up. Sorry, I screwed that up. Let me go backwards. Big, whoop, come on, there we go. Big B, big B, big S, little s. There we go. Blue snoring dog. Big B, little b, big S, big S. Big B, little b, big S, little s, okay? And hopefully you can see that I'm getting that by just combining these with that one. And you would go all the way through. So for this one, I would take this and combine it with that. Big B, big B, big S, little s. And you could go all the way through. And hopefully you can see that you would have 16 different squares here that you would have to fill. Hopefully you can also see that this is a pretty cumbersome way to figure out your possible outcomes and your possible genotypic and phenotypic ratios. Fortunately, there's an easier method, and that easier method is called branching diagrams or branch diagrams, okay? So let's use branch diagrams to figure out this dihybrid cross. So remember that our dihybrid cross in our scenario here is that we're going to take a mom who's heterozygous for her blue coat, and she's heterozygous for snoring, and we're going to cross her with a dad who is also heterozygous for his blue coat, and he's also heterozygous for snoring. Now, what I wanted you to do in order to figure out how to make this a branch diagram problem is I want us to consider each of these genes, the gene for coat color and the gene for snoring. I want us to consider these independently, okay? So it might even help you to draw 2 little Punnett squares. And that's what makes this easy. Punnett squares are easy when you're dealing with just one gene, right? So here we go. Let's just consider coat color first. Big B, little b for mom. Big B, little b for dad. And let's consider snoring. So big S, little s for mom, big S, little s for dad. Okay, hopefully you can see that for each of these, there are 3 possible genotypes. So let's make a diagram using this information. Let's just start with our coat color. 3 possible outcomes, right? Okay, you can be big B, and you actually have a 1 out of 4 chance to be that, right? You can be big B, little b, and you have a 2 out of 4 chance to be that. And you can be little b, and you have a 1 out of 4 chance to be that. Okay, so all I did was I took each of these options and for each option, I just made a little tree here, okay? Let's repeat that using this information, but you want to repeat it for each of these branches. Here's what I mean. So I want to draw 3 branches here. I want to draw 3 branches here. And I want to draw 3 branches here. Are you with me? Now, let's do the same thing. We have 3 options here. You can be big S, and there's a 1 out of 4 chance that that's the case. You can be heterozygous, 2 out of 4, or you can be this, and that's 1 out of 4. Okay, you guys get where I'm going. I'm gonna do one more here for you because I want to finish out this problem to show you something. And then doot, 2 out of 4, and then 1 out of 4. Now, here's the really cool thing. We can get a lot of information now just by following these branches. Here's what I mean. Let's take this big B and let's couple it with this branch here. So we're going to have a genotype of big B, big S, big S. We can even tell how many of these we would get. Remember how there were 16, 1, 2, 3, 4 by 4, 16 different squares that we would be filling in here if we did this longhand? Check this out. 1/4 times 1/4 is, whoops, come on back, guy. Hang on, sometimes this gets stuck. There we go. 1/4 times 1/4 is 1/16. So if we were to fill out our Punnett square, one of them would have this genotype here. Let's do this again. Take these 2 big B's and let's combine them with this branch here. Big S, little s. 1/4, 2/4, right? That comes out to be 2/16. 2 of our squares on our Punnett square would have looked like that. And let's just go through and finish filling this in. Big B, little s. Oops. That's 1/16. That's going to be 2/16. And then this is going to be boop, 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 boop. That's going to be 4/16. Here's how I got that. Look, 2/4 times 2/4 is 4/16. Let's do this one. Big B, little s, little s. That's going to be 2/16. Now let's do this last one. I'm going to combine these out. All right. And then this last one, teeny weeny guy here. Now, if I've done this correct, we should have 16 different genotypes that would have filled in, right? So we'll have 1, 2, 3, 4, 5, 6, plus 4 here. That's 10, 11, 12, 13, 14, 15, 16. This equals this mess here. This is easier to see. We can also get our genotypic ratios, 1 to 2 to 1 to 2 to 4 to 2 to 1 to 2 to 1. This makes this so much easier, okay? Let's use this method. I used this to get probabilities just now. Okay, so the probability here of getting a big B, big S is 1 out of 16. I also used it to get a genotypic ratio. Sorry, guys, it's hard to write on my tablet. And to get that, just get rid of your denominators, 1 to 2 to 1 to 2 to 4 to blah, 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 okay? Let's use this to get our phenotypic ratio. So let's go back. I'm gonna write up here what our parents look like one more time. Okie doke. So once again, if you get confused, what you want to do is you want to break it down by gene. So here's my 2 Punnett squares, boop, 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 right? Big B, big B, big B, sorry, big B, little b, boop, boop. Y'all know what I'm doing here. Now let's look at the possible phenotypes for each of these. In this Punnett square, there are 2 physical appearances. You can be blue or you can be gray, right? Hopefully you see how that's the case. There are 3 different ways to be blue. You can be this one, or this one, or a 3 out of 4 probability. And there's 1 way, or a 1 out of 4 probability to be gray. Over here, you can be snoring, or you can be a quiet dog. Trust me, that is not my dog, okay? You have a 3 out of 4 chance to be a snoring pooch and a 1 out of 4 chance to be a quiet pooch. So let's look at our phenotypic possibilities using these branch diagrams. So let's start here. You can either be blue or you can be gray, right? And you have a 3 out of 4 chance of being blue, and you have a 1 out of 4 chance of being gray, okay? For each of these, you can either snore or you can be quiet. Make this one a little shorter because I'm going to run out of space to write here. Snore, quiet. And again, 3 out of 4, 1 out of 4, 3 out of 4, 1 out of 4. Now, let's figure out what our options are here. You can be a blue snoring wine. Actually, you know what I'm going to do? Well, yeah, blue snoring wine. Okay. And, ooh, hold on. Sorry. Oof. There we go. Sometimes my tablet goes a little haywire. Okay, so I'm going to multiply this times this. This works out to be 9 out of 16. Okay. You could also be a blue quiet wine. So I'm going to put blue quiet. And that works out to be 3 out of 16. Hopefully you see how I get this. Blue snore, 3 times 3 is 9, 4 times 4 is 16. Blue quiet, which is this guy, blue quiet. 3 times 1 is 3, 4 times 4 is 16. Let's do this one. Gray snore. One times 3 is 3. 4 times 4 is 16. And a gray quiet. One times 1 is 1. 4 times 4 is 16. So you have a 9 to 3 to 3 to 1 phenotypic ratio, okay? And all of these add up to 16, right? And you can even tell what the probabilities are. A 9 out of 16 chance that you'll be a blue snoring dog, a 3 out of 16 chance that you'll be a blue quiet dog, etc. This here should equal, if you go back here and you count up how many of these guys are blue snoring and you add up the numbers, these should be equivalent, right? But this is an easier way to get at your phenotypic ratios, okay? Let's look at 3 genes, okay? You saw that it got a little tricky with 2 genes. What happens with 3 genes? Well, first of all, if you have 3 genes, let's just add another gene in here. So we've got coat color, and we've got snoring, and I'm going to add another fake gene in here. Wimes are gaseous. They can clear a room with their gas. So we're going to say that big G is super gassy and little g is not gassy. Okay, so I'm not sure that this dog actually exists, but for the purposes of our demonstration, let's say. Well, remember when we made our gametes before and I was like, okay, big B, big S, little s, big G, little g. Somehow we have to figure out, okay, this is mom, what are all her possible egg combinations? With 4, it wasn't too bad, but can you imagine, like, if you had to figure out all the possible egg combinations for the gametes for mom and dad? That by itself is tricky. But we can apply this idea of branch diagrams even just to figure out what gametes are. So let's just take each of these one at a time. Mom she can donate either a big B or a little b to each egg that she makes. Then she can do a big S or a little s. Then she can do a big G or a little g. So to figure out what her possible gametes are, all we have to do is follow the branches. So here's one branch, B, big S, big G, B, S, G. Let's look at this second branch here. Big B, big S, little g. All right, now, so we've exhausted this one. Let's move on. Big B, and we actually exhausted that one. Big B, little s, big G. Big B, little s, little g. Okay, we've exhausted that one, and we've exhausted that one. And we've actually exhausted this one now. Okay, big B, big S, big G. Oh, sorry, little b, big S, big G. Little b, big S, little g. So now we've gone and done that. And let's see here. Little b, little s, big G. Okay, so I've done that one. Little b, little s, little g. Now I've done that one, and that's 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7, 8. Excellent. So this helped us figure out this tricky problem. Once you have this information, you can plug it into a new branch diagram. So let's do this. Let's assume okay, so as I was saying, once you have this, you know all the possible gametes. Now, what if we wanted to do a branch diagram and figure out our phenotypic ratios? Let's just say that we have a mom that looks like this and a dad that looks like this, okay? This looks like a crazy problem, but it's not. It's easy to break down. So first, let's tackle each of these individually as an individual Punnett square. Right? And you guys know how to do this. I'm going to shorten this process because hopefully by now you know that you have a 3-to-1 ratio. This one will be blue. This one will be gray. It should be 3 out of 4 or 1 out of 4. For snoring, hopefully you know you have a 3 out of 4 versus a 1 out of 4 for quiet. And if you did this here, you would find the same thing. Okay, you would find that for gassy, there would be 3 out of 4, and for not would be 1 out of 4. Okay, so we can make a branch diagram. All right, let's start out with our coat color. You can be blue, right? And that's 3 out of 4 dogs, or you can be gray. And that's 1 out of 4 dogs, okay? Now you can snore, or you can be quiet. And so that's 3, 1, 3, 1. Okay, now for each of these, you can be gassy or not. Okay, and again, 3, 1, 3, 1, 3, 3, 1. Okay, so now we can figure this out. You can be a blue, snoring, gassy dog. And if you multiplied this out, you would have 3 times 3, which is 9, times 3, which is 27. I do that math right? Out of 4 times 4 times 4 is 64. Okay, I hope I did that math right. And you would go through and you would repeat what we've done before to get all of your phenotypic ratios. Hopefully you see how you could use this exact same method. Oops. I somehow had the same slide on here twice. But hopefully you can see how you would use the exact same method to figure out genotypic ratios, okay? So I've gone through all the information that you need to complete this lab assignment. Using this method, you can pretty much figure out any genetics problem that Dr. Bauer is going to give you. So what I've done is I have posted on Blackboard, you'll see the link to this video as well as a PDF to your assignment. It is a series of problems in which you will use these branch diagrams to answer a set of questions, okay? These problems are going to be due next week when you come, or right before your normal lab period. So if you have a Monday lab, they're due on February 15th. If you have a Tuesday lab, they're due on the 16th. If you are not coming to the in-person lab, because next week will be a group A week, and I will remind you of that because I know we're off schedule. But if you're not coming to lab in person, you can simply either email me your completed document, a PDF of your completed document, or you may turn a physical hard copy in at the main biology office. If you are having trouble with this assignment, email me and I will set a time to Zoom with you. If it looks like there's enough people having trouble, then I will set up a time to have a virtual tutoring session to help you with these branch diagram problems, okay? Hopefully you have found this useful. I think these are really fun. Treat these as fun little logic puzzles, and I think that you'll have actually a good time doing this assignment. Talk to you soon.